So welcome again. So we will start by the talk of Professor Rossmann. And you see the title on the screen. Please, Professor Rossmann.
Well, just once more, I really would like to thank the Biophysics Committee of the Sackler Award for making this possible. And I will be, can I be heard? All right, I'll try to stay here. I'll be giving you now a sort of scientific history of my own life in a relationship to virology. As you will see, I've been very much involved in developing technology for determining the structure of viruses and understanding how viruses work. And in doing that, I need to talk about not only what I did, but all the things which went around me in the time before and during my life, which made it possible. Obviously, nobody ever works alone in science. Science is always a question of collaboration and convergence, as we heard, of a variety of ideas and people. So I wonder whether there's a pointer here. Do we have a... Well, maybe I can point it. Sides. There we go. It's not very clear, but not sure that is. Oh, it's green now. Good.
This is better. The one on top.
Oh, that's much better. Thank you. This whole story, in a way, starts in Germany, in Munich, in 1912. Again, it's a question of a lot of different people coming together. The X-rays had been discovered in Munich by Röntgen some years earlier. And there was a very strong physics group in Munich at that time. It was Peter Ewald who really got things started by showing that X-rays could be diffracted by crystals. And he worked out the mathematics of the diffraction effect, leaving us today with what crystallographers know as the Ewald sphere. It also meant studying what crystals are, namely the repeat of some object in 3 dimensions. But we will not now have a lesson in crystallography. These ideas of Ewald were soon incorporated in the work of William and Lawrence, William the father, Lawrence the son, Bragg in England, in Manchester. The Braggs had come back from Australia. They had been, Lawrence Bragg was born in Adelaide, in Australia, and they come back to England. And it was actually Lawrence Bragg, when he was a student in Cambridge, who suggested the Bragg's law. Now, Bragg's law is really the same thing as had been looked at with Peter Ewald by von Laue, and regulated the idea, understood the ideas of how X-rays are diffracted from planes in a crystal. What that discovery leads directly to what I will be telling you about, as all very much connected. Kathleen Lonsdale was actually, she an English Quaker, and first woman to be elected to the Royal Society in England, was able to use X-rays to show that things like benzene ring is a planar ring, as had been suggested by, for instance, Kekule. So there was a connection here which related organic chemistry with the structures which crystallographers were starting to discover. Kathleen Onsdale was also a very significant person in my life, in that she was a member of the board of the school I was at, and she gave tickets to senior scholars in the school to attend the annual Christmas lectures in London. And I was one of the chosen people to go to these. And later, my relationship to Kathleen Lonsdale was very important in bringing me to Robertson and my introduction to crystallography. J.D. Burnell, an Irishman, was also an early person in Cambridge, looking at the structures of natural products in Cambridge, understanding crystallography and how to use it, looked at the structure of steroids. And Asbury in Leeds in England, notice all these people are from England. He was in the textile industry and looking at fibers. And he showed that the diffraction patterns of proteins, such as wool and nail from fingernails, had certain patterns. He classified these patterns into alpha and beta patterns. And later, particularly with Linus Pauling in California, his Asbury's alpha and beta patterns are related due to what we now know as the alpha helix and the beta sheet. Dorothy Crawford Hodgkin worked with Burnell in Cambridge, and again, she was a very important influence in my life. And she later got she worked on, for instance, the structure of penicillin, very difficult structure to solve. It had about, 20 or 30 carbon atoms. And she later got the Nobel Prize. I think probably the first Nobel, well, that's not true. Burnell got Lawrence Bragg got the Nobel Prize, and so did Dorothy Hodgkin for her work on vitamin B12. And then Monteith Robertson was another person who developed things like the isomorphous replacement method for small compounds, heme-like compounds, things which are at the center of hemoglobin, which I'll be talking about. He was my mentor and major professor in Glasgow, who had set up a well-known school of crystallography in Glasgow, and the structures, this was all leading to what later is what I will be talking about. And then here we have Burnell again, together with his American postdoc, Frank Pfannkuchen, who was able to determine roughly what the overall order is in a virus. Now, at that time, viruses really, or what the definition of a virus was at that time, was an infectious material which can pass through the smallest pores, smallest filters known at that time. And he looked at a plant vase, tomato bushes, tobacco mosaic vase, and he showed what the overall structure of this is, Burnell and Pfannkuchen together. And again, Dorothy Hodgkin, Crawford was her maiden name, together with Charles Spun, looked at penicillin, the first antibiotic, which was very important during the Second World War, saving many people's lives. Finally, Byfoot, a Dutch crystallographer, was able to use an effect known as anomalous dispersion to determine the absolute hand of chemical compounds, which Fischer in Germany had recognized that the carbon atom has a hand, the left and the right hand, which are related by a mirror plane or center of symmetry. And he, all the chemistry up to that point, nobody knew what was correct or wrong, but the Fischer convention suggested which way we would name things, left or right, according to how the light was rotated. He used crystallography, Byford used crystallography to determine what the absolute value of that is. And that will come back later as we look at the structure of proteins. Continuing this story, we have Linus Pauling in Caltech in California, who was starting to look at the structures of small peptides, small amino acids which form proteins, and looking at the bond which connects one amino acid to another, known as a peptide bond. And from that, as I mentioned already, he deduced structures which were helical and sheet-like, which he recognized would give rise to the kind of X-ray diffraction pattern which Asprey in England had classified some years earlier. Then Francis Crick and Jim Watson in Cambridge. Watson, as you probably know, was an American postdoc. Francis Crick was an Englishman. They were working together with Max Perutz, who I've also mentioned, in the physics department of Cambridge called the Cavendish, except that by the time they worked there, the then Cavendish professor, Sir Neville Mott, decided to push them out into a little hut, which I shall show you, because he thought this, what they were doing, was not really physics. And so that always happens to significant, usually happens to significant discoveries. They get pushed out, and nobody thought it was very important what they're doing. And they, of course, discovered the structure of DNA, which was maybe, in my opinion, the most important scientific discovery of the 20th century. They follow this up with a discussion of what viruses might be. The DNA structure had given them the idea of the genetic code of a sequence of bases along the DNA given you being translated into proteins. And so they recognized that that in a virus, which all they knew was a sort of a spherical ball, would have the genetic material, DNA or RNA in the inside, that the amount of genetic material in the typical virus, as was known at that time, the amount of information there was insufficient to code for a protein which completely covered all the genetic material. Therefore, the code surrounding shell of protein on a virus must be made out of many copies of the same protein. And so if a protein is associates with other proteins, and if each protein has the same structure, then each protein must have the same environment. And so the question was then, how can the proteins on the surface of a virus, each protein being identical, having the same identical environment, what kind of structure would that be? And that the Greeks had thought about that into the Platonic solids, regular polyhedra with identical faces. And these are, for instance, the tetrahedron here, and octahedron, and the biggest of these is the icosahedron. And on the icosahedron, you have 60 identical structures of a protein, or identical structures, whatever it is, making the same, each protein, each subunit having the same environment. So they suggested this would be the most efficient way for nature, for a virus, to have organized the identical proteins coded for by the code to be. And as it turned out, this was right, and was kind of verified by Casper and Klug, again, Don Casper and Aaron Klug in Cambridge. They were the first to take a crystal of a virus. It was tomato bushy stunt virus, and they were able to get the X-ray beam going in a certain special direction. And they found these strong, this is the diffraction pattern, that's a kind of, this is the diffraction pattern, it's a simplification pattern on the right-hand side, and there are certain directions which had strong reflections, which they showed would be along lines of symmetry. And in an icosahedron, you have different symmetries, 5-fold, 3-fold, and 2-fold, and they have well-known angles between them, about 20 degrees between 2-fold and 3-fold, and about almost 30 degrees between 3-fold and 5-fold. And these lines were consistent with this being an icosahedron. So they were the first to verify the idea that a virus, at least this case a plant virus, was indeed icosahedral, as had been predicted by Watson and Crick. Now, about this time that Watson and Crick were talking about these things, I was doing my PhD in Glasgow, and I was given by Robertson, my major professor, as crystals of a small organic compound, and I was asked to get the structure. And here's my first structure ever of a crystal structure. As you can see, the about 10 or so carbon atoms. There's a center of symmetry here. So all you had to do is to determine the structure of this. But this was a major achievement at that time. I went on to determine the structure of 2 more such compounds before obtaining my PhD. The interest in these compounds were not the structure itself, because the chemists already knew the structure, but rather the distance between the atoms. You can see the atoms individually, the electron density contours, and what is the distance of between a carbon-carbon single bond, a double bond, triple bond, and these are partial double bonds, 1.5 double bonds, and you could plot these and see the relationship between distances in atoms, angles in the atoms. That was quite novel at that time. We kind of take it for granted today that everybody knows this. In Cambridge, doing this, well, that work I just described I did in Glasgow, and I've already mentioned earlier today that I then went to Minnesota, where I learned something about computers for 2 years, and there solved some natural plant product compounds, terpenoids, and then came to Cambridge. And in Cambridge, I found a very different kind of atmosphere. It wasn't just doing experiments, but people were talking about all kinds of interesting things, maybe politics, but also science. And this was the hut, which I've also mentioned behind here, where Perutz and John Kendall, Francis Crick, and others. Max Perutz was, this is Max Perutz, was the head of the lab. His office was there. This office here was the office of Francis Crick and Sidney Brenner. All these, of course, were later Nobel Prize winners. And every morning at 11 o'clock, we would meet in the hut in the store. This is the door. And we would, Crick was there, Francis Crick was there, and we discussed all kinds of things. It was an awakening of my mind, working in this environment. My job was working with Max. He had collected a lot of diffraction data on hemoglobin, and I had to write programs and develop methods for solving the structure. We eventually produced electron density map such as this. And you can see here these long pieces of density, which we soon recognized to be alpha helices. I should have mentioned that John Kendall, a student of Max Perutz, had earlier determined at for the first time for any protein, the 6 angstrom resolution structure of myoglobin. And with Max, I was working on hemoglobin. Both these are oxygen-carrying proteins, hemoglobin in the blood and myoglobin in muscles. And when in 1959, we had this map of hemoglobin based on the programs I had written and developed the methods. And these methods continue to be a major way of solving proteins today, isomorph replacement. But there are other methods, as I will show you, which have sort of, in a way, superseded. It's now possible to supersede these methods. And here is then the structure of hemoglobin. Max Perutz took these maps and cut out shapes on each section. I showed you one section on the previous slide, and put them together. And you can see these long helices there. So it turned out that hemoglobin was had about 8 helices. There are 4 chains, the alpha chain and the beta chain, 2 of each. And that also gave an understanding of the Bohr effect, how the oxygen associated with the heme group. The heme group is, can't see it now, here, the heme group, the oxygen is, the heme group is the carrier of it, and how that is controlled by the structure. And that was clear from the structure, and Max Perutz really interpreted this. And this is then taking the hemoglobin structure apart to alpha chains related by a twofold axis to beta chains, one goes upside down. And the amazing thing here, and that really to me was an incredible discovery, that the structure of the alpha chain, the structure of the beta chain, and the structure of myoglobin, other oxygen carrier, this is myoglobin, the same chain, they all had similar structures. We didn't know at that time what the amino acid sequence was. That is later. But this was really the first time that we could see, anybody could see evolution at a molecular level. How did evolution come about? How is it that whales and humans have so much common in their organs which make them work? Because here we have a similar evolution at the molecular level, and it's the molecules, of course, which make the total organism work. I could say so much more on this, but really don't have time now. Another aspect of what we did at that time, particularly in John Kendo's group, there were these alpha helices. Oh, at that very same time, John Kendo's group in Cambridge was working on the higher resolution structure of myoglobin. Max and I and others in group Max had worked on the low, it was a 5.5 angstrom structure of hemoglobin, but Kendo with Dick Dickerson and others at David Phillips had worked on the high resolution of myoglobin, and there these rod-like structures could be seen. And this is a look at the rod-like structures and shows the regular repeat along the rod-like structures, which were the amino acids. And so this then verified for the first time the presence of alpha helices. What Pauling had predicted were indeed in hemoglobin. That was correct. Now, all this work in 1959 was very stimulating, seeing the relationship between the alpha chain and the beta chain in hemoglobin. And that gave me the idea that maybe one could find that relationship without actually going through all the isomorphous replacement with heavy atom structures. Together with David Blow, who had been a student with Max Perutz and had been in America when all this was happening, but he came back to Cambridge, I wrote, evidence is accumulating that many of the larger protein molecules are made up of identical or closely similar subunits. The reasons for expecting this from the protein part of virus structures were set out by Crick and Watson, as I've explained to you, and the prediction has been amply confirmed in crystallographic studies of spherical viruses. That's the work of Klug and Casper. And that gave me the idea that maybe one could use this, what we call now non-crystallographic symmetry, the relationship between the alpha and beta chain, to solve the structure without anything else. That I later gave the name of molecular replacement, was mentioned already. And today, molecular replacement is the method which solves structures of viruses, of proteins, things much more complicated than hemoglobin. When I say the methods, the percentage of structures which are deposited annually in the protein data bank is there are now 100,000 protein structures in the protein data bank, and the more than 3/4 of those structures which are deposited annually now are dependent on molecular replacement as the method of solving those structures. And those clearly a very significant part of our biological knowledge, knowing these many wonderful structures, many of which have earned other people the Nobel Prize because of the importance of the structure. Now, continuing about the history of this whole subject, we have David Phillips using, again, the same sort of methods, looked at the structure of a very small molecule, enzyme, lysozyme, and that was the first enzyme to be determined, and therefore once you look at the structure, you can also analyze what makes this structure, this enzyme, work as an enzyme, what makes it catalyze chemical reactions. In Purdue, by this time I got to Purdue, and I had to think about what to do in Purdue. I wanted to work on viruses from the very beginning because of their symmetry and therefore its relationship to the possibility of using what I was developing at that time, molecular placement. And I should mention at that time when I suggested the ideas incorporating molecular placement, it was not a popular idea at all. But I wanted to work on such things. So I couldn't work on viruses because it was much too considered to be much more too difficult. There would be no grant money. So I decided to work on enzymes which had symmetry. And I worked on lactate dehydrogenase to start with because it was a tetramer and would have symmetry, similar to some extent what happens in viruses, and that indeed was true. So we worked on, and I also chose lactate dehydrogenase because I thought it might have evolutionary relationships to other enzymes, and that turned out to be true. The NAD binding component of lactate dehydrogenase domain turned out to be a domain. We came to call it a domain. There was a NAD binding domain and a substrate binding domain. And this domain was soon, we discovered it in another dehydrogenase, aldehyde 3-phosphate dehydrogenase, and then Carl Brendin in Sweden looked at alcohol dehydrogenase, Len Banasek in Missouri looked at malate dehydrogenase, and we soon discovered that this structure binding NAD was present in many enzymes, and these enzymes were mostly important for getting energy, an essential component of life to convert structures, chemical compounds into energy and do something with the energy. So I thought that this was a very important structure, and here it is. It contains 6 parallel strands in the beta sheet with return alpha helices, and this structure is indeed the most common kind of what we now call fold in all the data strands. You can recognize it even if you haven't looked at a 3-dimensional structure because it has certain amino acid sequence characteristics where the alpha helices are formed, beta sheets are formed. So it turns out to be a very common fold. With that discovery, I thought would now be possible in Purdue to look at viruses. I was very fortunate at Purdue to have a lot of greenhouses where we could grow plant viruses, and plant viruses, you can get very large quantities, gram quantities of it. We could do what we liked, and we soon, it was not till 1970 that we were able to determine the structure of a plant virus. Steve Harrison, working in Harvard, had also been working on a plant virus. He had been in Cambridge working with Aaron Klug, and that's where he got that idea, worked on tomato bushy stunt virus. And so it turned out that the structure virus which we determined in Purdue had a very similar structure to what Steve Harrison had determined in Harvard. And we have here in yellow, we have an icosahedron, and as I said before, an icosahedron has 60 identical units, but also I should have mentioned that Casper and Klug recognized that many viruses are bigger than was possible from icosahedron, and they invented a system of quasi-symmetry. This is called T equals 3, or triangulation number of 3, and each of the 180, 3 times 6 is 180 subunits had this fold, which for some reason which I don't know has now been called a jelly roll fold. And this fold then subsequently turned out after we finished with the plant viruses, we then turned to animal viruses. We looked at common cold virus because I got to know Roland Ruckert, who had been working on picornavirus, small RNA viruses. And with him, we looked at a common cold virus. There are many common cold viruses. They're all somewhat similar. And we determined the structure of the first common cold virus, first animal virus, in fact. And it turns out, fortunately, that here is this common cold virus. It is organized in exactly the same way, and the subunits have again the jelly roll fold. So in the case of the plant virus, we have 180 of these folds organized in the way shown here. The triangle here is the asymmetric unit. There are 3 of these folds in one asymmetric, icosahedral asymmetric unit, 60 icosahedral asymmetric unit in the virus. And in the case of the picornavirus, the common cold viruses, we have, again, exactly like here, 3 different proteins in the asymmetric unit. They each have the same fold, or similar folds, at least. You can see here by comparing these folds. The differences in the plant virus, they're identical. In the animal virus, they have quite different amino acid sequences. So this was from the evolutionary point of view extremely interesting. Again, it was an extension of the earlier work on the hemoglobin. The structure itself of a common cold virus was also very interesting. It had a deep, what we came to call canyon. I had taken my family down the Grand Canyon in Arizona the previous summer before we solved that, and so it reminded me of the Grand Canyon. I call this the canyon. It's now generally known as the canyon. And together with Roland Ruckert and his student Barbara Sherry, they had looked at some neutralizing antibodies to the virus, and they could see that the virus was not neutralized when certain mutations happened, and these mutations occurred in different places. So one of the groups of antibodies all had mutations in this area, and that would be called the neutralizing immunogen at 1A, 1 because it's on the blue subunit, VP1, viral protein 1, and so on. And so we knew that these changes in the virus were on the surface where neutralizing escape mutations could occur on the surface of the virus. And that gave rise to what we call the canyon, or other people started calling the canyon hypothesis, where we now knew that the antibodies, neutralizing antibodies, bound to the surface of the virus, where I just showed you on these surfaces, but could not, because by this time Roberto Poliak at Johns Hopkins had determined the first structure of a Fab component, and therefore we knew that's too big to go into the canyon, and we knew that the residues on the surface of the virus where the Fab bound were very variable. And so the canyon hypothesis suggested that maybe the receptor for the virus, the molecule, cell surface molecule to which the virus would bind, would bind to the base of the canyon. And that is what actually turned out to be the case. And this, again, was now the first use of electron microscopy. We couldn't crystallize this, and so we were able to show that the receptor, which was later discovered to be ICAM, intercellular adhesion molecule 1, bound as predicted into the canyon. Oh, I thought there was one. Well, never mind. Another discovery which we made is that certain compounds bound into a hole inside the viral protein 1, and this is one of these compounds. And these compounds had the ability to neutralize the virus, and we could understand how this happened. And I don't have time to explain this now because I see the time is advanced quickly. But these compounds led to the development of an antiviral compound called pleconazole, which was very effective against common cold in phase 1, 2, and 3 trials, but it was not given a license by the FDA because it had it could make women on birth control hormones could they could then conceive in the presence of, if they were taking this gluconal antiviral compound. Nevertheless, the compound can be and has been used in compassionate case uses, and here the successes of some time ago, I don't know what the score is now. And then going further, where this was early success on viruses, but in terms of structural results, Hartmut Michel in Germany, together with Robert Huber, was able to determine the first structure of a protein which lives in a membrane. There are now many membrane proteins are known. These are not soluble, and I also very large DNA protein viruses, and I'll say more about that if I get time, could be determined. Stites, Ada Yonat from the Weizmann Institute, and Venki Ramakrishna all determine the structure of ribosomes, the machinery which translates the DNA into RNA into proteins. And then also Richard Kuhn at Purdue, together with myself, were looking at lipid-containing viruses, much more complicated than the Bicona virus, common cold viruses. And here is an example of dengue virus, which is flavivirus, an immature virus and a mature virus. They're different, and what happens when the virus matures. I'm going very fast at this point, and I think I'll just pass over this. We had to combine here crystallography together with electron microscopy, because we couldn't really usefully crystallize these viruses, but we could crystallize and get the structure of the components of the virus. So this is an electron density map of the virus, which we obtained in Purdue. And we knew from work which had been done by Felix Ray in Steve Harrison's lab. Felix is now at the Pasteur Institute. And they had determined the structure of the surface protein, a glycoprotein. We knew the structure of that, so it was possible to fit this to the electron density. This is the structure which had been determined by Felix Ray, and it fits very nicely. For tick-borne encephalitis virus, actually a homologous virus, and this fits there also. So this gave us a structure of the dengue virus. And this dengue virus actually belongs to the same family of viruses as Zika viruses, something about which I spoke here yesterday, and that actually was very important for determining the structure of Zika virus earlier this year. And again, I don't have time really to go into this, but we were able to follow how the virus matures into a mature virus by looking at the structure of the immature virus, and then of course the virus becomes fusogenic, and we have the structures, all that. Yeah, I'll skip over this. And now I've introduced electron microscopy, and this we used it initially in combination between crystallography and electron microscopy. Electron microscopy could look at bigger viruses, bigger components which you couldn't crystallize. But in recent years, last 3 or 4 years, electron microscopy has advanced rapidly, both in how to use the data and how to collect the data. With direct counting electron counters, we can now use electron microscopy to directly determine the structure at near atomic resolution. And that is actually what we did with Zika virus and have also done with certain other viruses. And the methods of combining, methods of now electron microscopy with the new detectors have been very powerful in determining all kinds of very fascinating, interesting structures which could never have been done using only crystallography. Another group of viruses which we examined are bacteriophages. And the bacteriophages, we are now developing bacteriophages as tools for medical tools. For instance, you probably all know that the antibiotics which we have in hospitals today are soon will be not being effective because the bacteria and other microorganisms are escaping from the currently available antibiotics, and therefore a virus which can kill a bacteria can be probably used as an antibiotic, can also be used in many other ways as a way of gene therapy. We've been looking at T4 virus, which is shown here. It has a head, just like the viruses, has icosahedral ends in the equatorial middle, has a tail through which the DNA is ejected when it lands on a E. coli host. But we are trying to develop these tools now for medical reasons, purposes, and now we have a look at this virus here. We determine the structure of the individual proteins crystallographically, and we put them all together with the electron microscope. And here's this T4 virus landing with its long tail fibers on E. coli surface. The E. coli is a bacteria. There's a shadow of the virus on the surface of the E. coli, and now it unwinds the short tail fibers, and they fasten well to this, and it winds like a drill, puts a tube down there and drills to the outside of the virus and pushes its DNA, the genomic material, in there. These are lysozymes which cut through the central cell wall, and that's how these bacteria work. The other viruses, Phages-174, which don't have tails, but actually make tails as they go along. Not enough time to explain that. And also I want to mention very large viruses, which have recently, the last 10 years, been discovered, particularly, for instance, starting with the work of Dieter Rald in Marseille in France. Large viruses have escaped us for many, many years because they're too large. They're so large they pass through filters, and therefore they've been thrown away. Nobody knew about them. And we've been looking at some of these. This shows how large they are. This is Atlas carrying a virus. This is a mimivirus, one of the first viruses discovered by Dieter Rald. This has a special vertex. We call it a stargate because it looks like a star. And also R.V. Minsky at Weizmann is working on this. This is the RNA, and I don't have time much to say much about this. But this is work of R.V. Minsky, in fact, how the stargate opens. You can see the inside coming out of the virus. These are these very large viruses. These large viruses also develop antibodies. It's in all waters. They're common. They grow on amoeba, and these large viruses are going to be discovered, I think, in due course to be major hazards for humans. Another virus which we're working on with Jim Van Etten from University of Nebraska, PBCV, Paramecium bacillus chlorella actually seems to be involved in causing schizophrenia as an example. So we were able to determine the structure of for instance, this PBCV structure here. Don't have time to give you details, but interestingly, the major capsid protein is related to the structure of common cold virus, and here we have the structure of the individual monomer of the capsid protein, and it makes hexamers like this, and these hexamers actually correspond to the ideas of Casper and Klug and how viruses are put together. And so we have a direct relationship between these big viruses, the evolutionary relationship between these big viruses, very large viruses like mimivirus, and the smaller, very small virus, Picona RNA viruses like common cold virus, like poliovirus, is also Picona virus. And for instance, these the structure of this large virus, which has two of the jelly roll folds in one polypeptide chain. Here, it's, for instance, occurs also in adenovirus. Adenovirus is really a large double-stranded DNA virus. It also occurs in other phage bacterial viruses like PRD1. and also in plant viruses. So again, all these viruses, not every virus, but by no means every virus, but a large group of viruses have had this common evolutionary origin, starting with the jelly roll fold. And in the case of PBCV, we thought it was icosahedral, but then we discovered that there is actually a spike at one end, and this spike is involved, as you shall see on the next slide, involved in its ineffectivity. In this cavity here, there seems to be an enzyme which is inserted to digest cell walls of the chlorella. And here we have these PBCV viruses, and you can see the special vertex here with the spike, and these are all pointing towards the cell wall, where the spike goes into the cell wall and ejects its enzyme to digest the cell wall. And I want to end up with thanking Max Perutz, who really taught me in many ways the first time what is important in science. And science is not just a nice game solving problems, but there's a reason for which we try to solve these. And this is Max with the model of hemoglobin, which he constructed. This is my group today, and I've had many people in my group. Over the years since I first came to Purdue, something like 150 postdocs and maybe 50 graduate students have been with me. I don't have time to acknowledge all of them, but these are some of the people working on the Bicorner viruses and on flaviviruses and on the large double-stranded DNA viruses and on bacteriophage. All this has happened in the lab since I first came to Purdue in 1964. Thank you.